Hi everyone, welcome to Purine and Pyrimidine Metabolism lesson. In this lecture, I will talk about function, structure and metabolism of purines and pyrimidines. I would like to also inform you about the biochemical mechanism of some disease related to purine and pyrimidines. Knowledge of structure and function of nucleotide is essential in understanding genetic problems and many aspects of some disorders, pathophysiology. And when you look at the nucleotide metabolism or nucleotide structure, nucleotides are composed of three subunit molecules. And nitrogenous base unit, we have to synthesize this unique base in DE-NOVO and SALVAGE pathway in purine and pyrimidine metabolism, and 5-carbon sugar ribose or deoxyribose, and a phosphate group consisting of 1 or 3 phosphates. And there are two purines, adenine and guanine. Also we have 3 pyrimidine molecules named thymine, cytosine and uracil for DNA and RNA synthesis in nucleotide or deoxynucleotide forms. Generally, we will talk about nucleotide metabolism. We are focusing on DNA and RNA, nucleic acid metabolism, deoxyribonucleic acid or ribonucleic acid. The discovery that genetic information is coded by a polymeric molecule composed of only 4 type monomeric unit called nucleotides was one of the most important scientific achievements in biological science. In the cell, not only DNA and RNA but also many molecules which have purine and pyrimidine structure are responsible for many important processes. Now let's look at these molecules. And ATP, GTP, NADH, FADH, adenosine methionine, UDP, glucuronic acid, CMP, GMP, and are accepted or have some purine and pyrimidine base or nucleotide, and some drugs such as anti-cancer or antiviral drugs also have purine and pyrimidine analogs and activated in the cell and has their activity during polymerization or transcription reactions. Also, I would like to give some information about human disease that involve purine and pyrimidine metabolism, such as Leishman syndrome, adenosine deaminase deficiency, uric aciduria, hyperuricemia, and good disease. And polynucleotide and DNA and RNA accepted that or has polynucleotide structure. DNA is made up molecules called nucleotides. Also, as I told you, each nucleotide contains a phosphate group, a sugar group, and nitrogen base. Also, 4 types of nitrogen base are adenine and thymine, guanine, and cytosine in DNA, but thymine must be changed to uracil in RNA metabolism. Of course, there are very important differences between DNA, RNA structure and function. I hope that we met another lecture, my lecture about nucleic acid genetic information, and we will talk detailed information about these metabolic processes, replication, transcription, and translation. And NAD or FAD, 2 cofactors that are involved in cellular respiration. They are responsible for accepting high energy electrons and carrying them ultimately the electron transport chain, and where they are used to synthesize ATP molecules. And these electron carriers molecules accept electrons from Scott cycle, glucose metabolism, or beta oxidation of lipids, and they are reduced into NADH and FADH2. And electrons of NADH and FADH2 must be moved to oxygen step by step, and we say electron transport chain in mitochondria inner membrane. And after that reactions, we have to synthesize an ATP, and ATP synthase is responsible for the synthetic way of ATP from ADP. It is another important lecture in biochemistry. Also, I will give detailed information in mitochondria, electron transport chain, and ATP synthesis in my next lecture. Also, as adenosine methionine is a sulfur-containing molecule, and it is well known as a metal donor for the major of metal transferase reaction, as including DNA and RNA metabolism. And another one, UDP glucuronic acid, an activated form of glucuronic acid, and need for synthesis UDP glucuronic acid, UTP. After that, we can synthesize UDP glucuronic acid for conjugation reaction in steroid and thyroid hormones, and catecholamine and estradiol metabolism. And it is one of the most important reactions for conjugation of many molecules in the cell. And another one, cyclic AMP and GMP, has hormone signal transport system. For example, epinephrine has one receptor which is inside of cell membrane, and when epinephrine binds to the receptor and activate adenyl cyclase enzyme, and enzyme produce cyclic AMP from ATP, and cyclic AMP has very important role in glucagon and adrenaline metabolism. Also, it is these molecules has many important roles in hormone metabolism. Another one is some molecules has purine and pyrimidine structure, and called anti-metabolite. In anti-metabolite, molecules similar to substrate of a key enzyme and inhibits this enzyme in very important chemical reactions. For example, is 5-fluorouracil is an analog of uracil molecule, has been associated with inhibition of thymidine synthase, is this key enzyme for synthesis of thymine nucleotide. Another one is methotrexate. Methotrexate is similar to dihydrofolate and inhibit dihydrofolate reductase. It is also another competitive inhibition, and also another key enzyme for or activation thymidine synthase enzymatic reaction. And acyclovir and lamivudine are chemicals, and we can use these purine and pyrimidine analogs for treatment of viral infections. Acyclovir is a synthetic nucleotide analog active against herpes viruses, and also lamivudine is another synthetic nucleotide, pyrinucleotide analog with activity against hepatitis B viruses. Another one is allopurinol. Allopurinol is a specific and unique inhibitor of xanthine oxidase and block hyperuricemia, and after that inhibition of xanthine oxidase, uric acid inhibition must be blocked. And human tissues can synthase purine and pyrimidine from amphibolic intermediates, ingested nucleic acid and nucleotide, which therefore are dietary non-essential, are degraded in the intestinal tract to mononucleotide form, which may be absorbed or converted to purine and pyrimidine bases in purine or pyrimidine degradative way. And after that, all of this reaction, we can obtain uric acid or beta-hydroxypropionate. For this reason, we have to synthase purine and pyrimidine nucleotides or deoxyribonucleic form of nucleotides inside of cell in synthetic way, synthetic way or salvage pathway. Now we are talking about purine nucleotide metabolism after general information about base or nucleotide purine and pyrimidines. And when you look at biosynthetic sources for purine ring system, purine ring system is built on ribose 5-phosphate. This molecule comes from pentose phosphate pathway, and purine ring system comprises 4 nitrogen and 5 carbon atoms. Ring system is numbered starting from nitrogen coming from aspartic acid. Now let's take a look at a source of carbon and nitrogen atoms of purines. You can see there's number 1 nitrogen derived from nitrogen group of aspartic acid, carbon 2 and 8 from formate group, and nitrogen 3 and 9 amide group of glutamine, and carbon 4, 5 and 7 came from glycine amino acid, and carbon dioxide give 6 carbon of purine ring system. Now we are going to synthesize this purine ring step by step. First step is in de novo synthetic way, in the purine de novo synthesis. We need ribose 5-phosphate, and ribose 5-phosphate is a unique substrate for PRPP synthase, phosphoribose phosphate synthase, and enzyme need ATP and transfer pyrophosphate group to sugar and produce PRPP. Enzyme substrate depend on pentose phosphate pathway. PRPP is used as substrate by a large number of phosphoribose transferase, such as hypoxanthine 1 phosphoribose transferase and adenine phosphoribose transferase. It is the first step of synthesis. PRPP is the active form used in purine de novo salvage and pathway in pure metabolism and pyrimidine synthesis. It is key and molecules for purine and pyrimidines, and its regulatory step for synthetic way. The enzyme is regulated by end or last product. Its inhibition or activation reveals a clinical picture. And the second step of purine de novo synthesis is depend on PRPP glutamyl amido transferase. It is another another important enzyme for purine de novo synthesis, because the enzyme makes 1 and 9 beta and glycosidic bond between the first carbon of the sugar and the nitrogen atom from glutamine. Glutamine transfer its amide group to PRPP to replace pyrophosphate and produce 5-phosphoribosyl amide. The enzyme PRPP glutamyl amido transferase is controlled by feedback inhibition like AMP, GMP, ADP or ATP, and we obtain phosphoribosyl amine and must be go to synthesis de novo Purine ring system step by step, and after that glycine amino acid enter the reaction and give carbon 4, 5 and nitrogen 7 atoms to ring system, and then methyl tetrahydrofolate, active form of folic acid, and give 8 carbon of purine ring system and reduce tetrahydrofolate. After that glutamine enter again the reaction and give third nitrogen atom of ring system, and after that we close the ring and produce amido-imidazole ring system. After that the carbon dioxide enter the reaction and give 6 carbon of purine ring system. Another amino acid, aspartic acid, enter the reactions and bind to ring system and give nitrogen atom and release fumaric acid. And after that we reach to last step of purine ring system, synthase of purine ring system, and formyl tetrahydrofolate, another active form of folic acid, give second carbon of purine ring, and after that we close this ring system and obtain inosine monophosphate. Inosine monophosphate has a unique role for synthase AMP and GMP. First way, AMP use AMP dehydrogenase, is one of the important enzyme for treatment of or targeting of chemotherapy, and produce XMP, and then we need 1 mole ATP and glutamine, and glutamine transfer to nitrogen atom to second carbon atom of a ring system, and we obtain GMP. And this is amination reaction. There's only one atom difference between AMP and GMP. Another way, adenosuccinate synthase enzyme use GTP, and aspartate, a substrate, another substrate, and aspartic acid enter a ring system, 6 carbon, binds 6 carbon and release again fumaric acid, and transfer 1 nitrogen atom to ring system, and we produce AMP. And AMP is 6 amino purine, and GMP is 6 oxy 2 amino purine. Also AMP synthase is called another amination reaction. After that AMP can go to GDP or ADP or ATP. GMP also goes to GDP or GTP. It is possible. And after that, when you look at feedback regulation, because we have a different type regulation mechanism for purine DE-NOVO synthesis. First is allosteric inhibition. Last products like AMP or ADP, GMP or GDP inhibits first and second reaction of purine DE-NOVO synthesis, and it is a very important way for regulation of purine system, and also ATP and increased synthase of GTP. Also GTP is responsible for increased ATP level concentration. This is positive regulation. Another negative inhibition or regulation called substrate products level inhibitors, like AMP and GMP have this kind of role, and is products AMP and GMP can be inhibit itself. It's another important regulative role way of regulation of DE-NOVO synthesis of purines. And when you look at degradation of pure nucleotides to uric acid, and it is very important metabolic area for us, because end of this reaction produce uric acid hyperuricemia. It's a very dangerous molecule for us. People are facing many problems because of hyperuricemia, and we have avoid high uric acid level in blood. End of all this reaction, we can produce uric acid or after that degradative way. When you look at this reaction, is AMP or GMP and other nucleotide monophosphate form or synthetic form of nucleotide can be used. 5' nucleotide, 5' NT enzyme can be determined as one phosphorylase enzyme, cut off one phosphate group from AMP and produce adenosine molecule, and adenosine go to inosine and used by unique enzyme, another unique enzyme, adenosine deaminase, and inosine can be used PNP, pure nucleotide phosphorylase, and turn to hypoxanthine. Hypoxanthine goes to xanthine and uric acid by xanthine oxidase. Another way, GMP guanine monophosphate can be used as substrate for 5' NT, and guanine monophosphate turn to guanosine, release one phosphate group from molecules, and we obtain guanosine, and guanosine goes to guanine and xanthine and uric acid. Both nucleotide, guanine or adenine nucleotide, must be go to uric acid in degradative way of purine metabolism. And there's a lot of problems in related to purine degradative way. We will look at what it is problem. And as I told you, uric acid is very dangerous molecule for us, and hyperuricemia is related to increased uric acid in blood, and there is some important disease associated with defects in purine metabolism, such as good and Lesch syndrome, kidney stones, and severe combined immune deficiency. Hyperuricemia is one cascade, and first of all is increased uric acid level, and uric acid combined with sodium urate molecule and turn to sodium urate, and this molecule turn to crystals, and this sodium urate crystal deposit in many tissue, and after that many reactions, and we met as a very serious inflammation. It's very dangerous condition for people. Uric acid is insoluble in urine and cause stone formation. As a result, acidic urine causes uric acid and calcium stone to form. And which problems are responsible for increased uric acid in blood, hyperuricemia? First is PRPP synthase enzyme increased activity. It's a genetic problem. If a PRPP synthase activity increased, we can produce the PRPP from ribose 5-phosphate. It is for first reaction of DE-NOVO synthase of purines, and goes to this PRPP must go to AMP and GMP, and then this molecule again go to the degradative way of purines and turn to uric acid, and increased PRPP synthase activity we face to hyperuricemia. And another problem is related to increased uric acid in ischemia and reperfusion. We know that ATP degradation is increased in ischemia condition. ATP turn to ADP, then AMP, adenosine inosine, and increased hypoxanthine level activate xanthine oxidase enzyme, and enzyme synthase xanthine and uric acid. And we have another serious problem during ischemia and reperfusion conditions. If xanthine oxidase activity increase in the reperfusion condition, enzyme can produce oxygen radicals and hydrogen peroxide. Hydrogen peroxide can be turned to hydroxy radicals. Ischemia, and during ischemia and reperfusion, we have two important pictures. First is increased uric acid level in blood. Another one is increased oxygen and hydroxyl radical, which are responsible for very dangerous problems in our body. And another problem related to carbohydrate metabolism in glycogen metabolism. Enzyme glucose phosphatase is responsible for production glucose from glucose 6 phosphate. It is key enzyme. You can block this enzyme. This substrate level must be increased. In this case, G6P must be go to hexose monophosphate pathway and produced ribose 5 phosphate. And increased ribose 5 phosphate is biochemical mechanism as similar to increased PRPP synthetase enzyme activity. In this case, ribose 5 phosphate increased and results to increased PRPP molecule and turn to and activate to DE-NOVO synthesis. And end of all of this stage, and you see is produce increased ribose 5 phosphate production and cause to here hyperuricemia. This results in an abnormal accumulation of glycogen in the liver, causing the liver to enlarge and producing symptoms of hypoglycemia. Children with a disease usually have poor muscle development and osteoporosis and suffer from hyperuricemia. It's very important problem for glucose metabolism, and we met increased uric acid level in blood. And gut disease, we have different type problems such as hyperuricemia, and chronic or episodic joint pain due to deposition of sodium urate crystals, and consequently inflammation. Is a very serious problem for patients, and as I told you, uric acid is minimally water soluble, and most cases of gut arise from inadequate excretion of by the kidneys, leading to build up uric acid and the precipitation of urate stones in the kidneys and extremities. You can see what it is a problem in extremities. And the joint of hands and feet are prone to accumulation of crystals because of reduced solubility of sodium urate and the slightly cooler temperature of the extremities. And we need allopurinol for treatment of a gut disease. When you look at purine metabolism, we have another important problems in a degradative way related to adenosine deaminase enzyme. We say adenosine deaminase deficiency. You know that adenosine deaminase use adenosine or deoxyadenosine as substrate and produce inosine. Inosine goes to hypoxanthine, xanthine, and uric acid. This is the degradative way of purine purine metabolism. But you blocked adenosine deaminase, deoxyadenosine or adenosine level increase in the cell, and this molecule must be go to step by step to deoxy-ATP. Deoxy-ATP is a very important inhibitor of ribonucleotide reductase enzyme. Is one of the key enzymes for DNA metabolism, because its enzyme use the phosphate form of nucleotide and turn to deoxy form and turn to deoxytriphosphate group for substrate of DNA polymerases. You blocked ribonucleotide reductase, you cannot produce deoxy form of nucleotide, and DNA synthase is blocked. And in this case, lymphocyte T and B lymphocyte is affected so badly in this condition. And when you look at ribonucleotide reductase, we are trying to figure out how it is work, because this reaction is very complex. When you look at nucleotide phosphate and so a substrate for enzyme and turn to deoxynucleotide diphosphate and turn to deoxynucleotide triphosphate, and the enzyme must be activated again and again, and for this aim we need another molecule such as thioredoxin, FADH and NADPH, and or glutathione reductase, glutathione reduced glutathione, and glutaredoxine and NADPH. You can see its reaction circle is very complex, but its ribonucleotide reductase is a key enzyme that mediates and synthesis of deoxyribonucleotase, DNA precursor, of course. It is key enzyme for polymerization DNA reaction for DNA metabolism. Adenosine deaminase deficiency is a genetic defect in adenosine deaminase gene, are among the most common causes for severe combined immune deficiency. Patients suffer from lymphopenia, severely impaired cellular and humoral immunity, and failure to have some problem in recurrent infections. But we have different type of treatment for these patients, such as bone marrow transplantation and enzyme replacement therapy and hematopoietic stem cell gene therapy. Another problem is in pure metabolism. It is a rare disease, hypoglycemia. In this case, it's also a genetic. It has a genetic problem in xanthine oxidase gene, and xanthine oxidase blocked. In this case, xanthine level or uric acid level decrease, and with xanthinuria and xanthine stones, it can be problems for patients. And idiopathic hypoglycemia usually requires no treatment for patients. When you look at another important synthetic way called salvage pathway, and we have to back to again purine degradative way, you remember that GMP or IMP or AMP must be go to uric acid. In these reactions, we produce hypoxanthine, guanine, and adenine base. And hypoxanthine and guanine can be used as very unique enzyme named hypoxanthine guanine phosphoribosyl transferase. enzyme use this base, hypoxanthine and guanine, and also has another unique substrate, PRPP, phosphoribosal pyrophosphate, because we need to product ribose and phosphate group from base, and we obtain IMP and GMP. After that, the molecule goes to GDP and GTP, or AMP, GMP, or ADP or ATP. It's very it's a good alternative way for us compared with de novo synthesis, because only one step, only one enzyme is responsible for production of nucleotide. It's very so big advantage for especially brain tissue. And if you blocked this enzyme, hypoxanthine and guanine phosphoribosal transferase, and in this case you blocked all products, and you cannot produce AMP and GMP, and hypoxanthine and guanine must be go to here uric acid, xanthine and uric acid, and in this case we have there is very dangerous problem for us called Leschnian syndrome. Leschnian syndrome is genetic disorders and depend on deficiency of hypoxanthine guanine phosphoribosal transferase, and result to failure salvage hypoxanthine and guanine to the corresponding nucleotides AMP and GMP, and this leads to PRPP accumulation, and again this PRPP goes to de novo synthesis and increase AMP and GMP concentration, and after that end of this stage we met excess purine synthesis. The excess purine and degrade to uric acid, causing increased blood levels of this molecule, hyperuricemia, and deposition of sodium urate crystals in the joint kidneys. When you look at as main problems in Leschnian syndrome, first is gut-like episodes of joint pain and kidney stones, and as well as severe neurologic problems, and also increased uric acid level, and phase 2 hyperuricemia. When you meet this kind of patients, you have to remember Leschnian syndrome, hyperuricemia and joint pain and kidney stones and some severe neurologic problems. And now we look at the pyrimidine nucleotide metabolism. Also, we have to follow same way. First of all, we look at source of carbon and nitrogen atom in pyrimidine ring system. As we look at this picture, we can see glutamine, carbon dioxide, and aspartic acid are a substrate for pyrimidine de novo synthesis. We cannot use glycine for pyrimidine metabolism. And when you look at this picture, nitrogen 1, carbon 4, carbon 5, and carbon 6 atoms come from aspartic acid, and carbon 2 in the ring system from carbon dioxide, and number 3, the third number of nitrogen from amide group of glutamine. Now let's go and look at how we can synthesize purine nucleotide step by step. First enzyme is carbamoyl phosphate synthase 2. Enzyme use glutamine and carbon dioxide and produce carbamoyl phosphate. As an enzyme catalysis, the first committed step in pyrimidine biosynthesis and also have positive allostatic effectors such as PRPP and ATP. Although UTP and UMP are negative allostatic effectors of enzyme carbamoyl phosphate synthetase 2. Enzyme is in pyrimidine biosynthesis. Enzyme serves as a rate limiting step and catalyzes following reactions, and we obtain carbamoyl phosphate and including one carbon and one nitrogen atom. And I want to give another information about key enzyme and name carbamoyl phosphate synthase and responsible for production of carbamoyl phosphate in mitochondria. Enzyme produced carbamoyl phosphate such as carbamoyl phosphate synthetase 2, but it is a key enzyme for ammonia or amino acid metabolism, but produce carbamoyl phosphate, and then it's a key enzyme for urea cycle. Carbamoyl phosphate synthetase 1 deficiency in an autosomal recessive metabolic disorder that cause ammonia to accumulate in blood. We know that very well ammonia is toxic if the levels become too high, especially nervous system is sensitive to the effects of excess ammonia. And in some case, you have some problem mitochondrial membrane. Carbamoyl phosphate leads to cytosol from mitochondrial matrix and enter to pyrimidine synthase. It is possible. And as I told you, first enzyme is responsible for nitrogen and urea cycle in mitochondria. Other one is in cytosol and produce also carbamoyl phosphate and use pyrimidine synthesis. And first enzyme is have a positive regulator such as an acetylglutamate, and also glucagon and cortisol increase protein synthesis as a positive regulator hormones, and also high protein diet has positive effector in for first enzyme. Also increase proton concentration has blocked enzyme activity, and second enzyme carbamoyl synthetase 2 in cytoplasm has positive activator ATP and PRPP, also has negative regulator UTP. Sometimes UMP has a role as a negative regulator for enzyme 2. And after that, this first reaction, aspartic acid entered the reaction, and enzyme aspartate transcarbamylase produce carbamoyl aspartic acid. After second reaction, we can obtain all carbon and nitrogen atoms for pyrimidine synthesis, and dehydroorotase close to ring system, and we obtain dehydroorotic acid is the first pyrimidine base, what we cannot use in any metabolic processes. And dehydroorotate dehydrogenase produce orotic acid. Enzyme is binds to mitochondrial membrane. For this reason, we said that pyrimidine synthesis in cytosol and mitochondria because of dehydroorotate dehydrogenase enzyme. After that, one phosphoribosal transferase enzyme. You remember these reactions. Looks like hypoxanthine guanine phosphoribosal transferase, orotate phosphoribosal transferase, transfer ribose and phosphate group to orotic acid and produce OMP. OMP has one extra carbon dioxide group, 6th carbon of pyrimidine ring system, and this carbon dioxide released from OMP and turned to UMP, uridine monophosphate, and we obtain UDP and UTP. So we need ATP, of course, and UTP is substrate for CTP synthase, and also enzyme use glutamine, and glutamine give one nitrogen atom to pyrimidine ring system, and we produce cytidine triphosphate. Another way, UDP goes to deoxy-UDP by ribonucleotide reductase. Also, you remember this enzyme, and we produce deoxy-UMP. Deoxy-UMP is a substrate for thymidylate synthase. An enzyme uses active form folic acid, methylene tetrahydrofolate, and produce dihydrofolate and produce TMP. There's only one difference. We can add one extra metal group to a ring system of uridine and obtain thymine nucleotide. It is one of the key enzymes in pyrimidine metabolism. I would like to look at this metabolic process a little bit deeply, and we can see deoxy-UMP is substrate for thymidylate synthase and produce deoxy monophosphate, and also enzyme use methylene tetrahydrofolate as another substrate and produce dihydrofolate. We have to regulate this dihydrofolate again to tetrahydrofolate because of activation of thymidylate synthase, and another unique enzyme, dihydrofolate reductase, and use NADPH and produce tetrahydrofolate, and certain amino acid give one carbon group to these molecules and release glycine and produce nitrogen 5 and 10 methylene tetrahydrofolate. We have to achieve this reaction again and again. If you block the dihydrofolate reductase, you cannot produce tetrahydrofolate, and in this case thymidylate synthase cannot find substrate methylene tetrahydrofolate blocked. And 5-fluorouracil is converted to 5-fluorodeoxy monophosphate and act as a suicide inhibitor of thymidylate synthase, and it is analog of uracil, is a combative inhibitor of thymidylate synthase. Another one is methotrexate, is a folate analogs and act as a potent competitive inhibitor of dihydrofolate reductase. Both antimetabolite can be used treatment of different type cancer, so it has a very important role in chemotherapy. And pyrimidine metabolism differs from purine metabolism. First is we cannot use glycine. Ring system is formed first, then ribose and sugar are added. In purine system, we need firstly one ribose phosphate, then add ring system step by step. It has relationship with mitochondria, but purine is in only cytosol. Also, we accepted that there is no special salvage pathway in purine metabolism. Of course, we have one enzyme, phosphoribosyl transferase or phosphotransferase, but this enzyme has key role in pyrimidine DE-NOVO synthesis. And how we can regulate pyrimidine DE-NOVO synthesis in prokaryotic cell and eukaryotic cells. In E. coli, pyrimidine biosynthesis inhibits by the last products, cyclophosphate, and this molecule inhibit second reaction of pyrimidine synthesis, and ATP is positive, has positive role of activation of pyrimidine synthesis. And another way in animals, UDP and inhibit first step of pyrimidine synthesis, and ATP activate this step. This is a regulation of pyrimidine DE-NOVO synthesis. Also, I would like to speak folic acid deficiency. Now you know very well folic acid is a key molecule for purine and pyrimidine synthesis because sources of carbon 3 and 8 in purine metabolism, and we need folic acid and thymine nucleotide synthesis. As blood levels of folic acid may become inadequate due to dietary insufficiency or poor absorption due to intestinal problem or alcoholism. And folate coenzyme concentration may also decline as a result of treatment with drugs that inhibit dihydrofolate reductase such as methotrexate. Patients with folic acid deficiency has different type symptoms, for example diarrhea, but basic or principal symptoms are weakness due to megaloblastic anemia arising from impaired cell division in the bone marrow. And you understand very well, if you blocked or decreased the folic acid levels, you have some problems in the thymine nucleotide synthesis or Purine DE-NOVO synthesis. Folate or folic acid deficiency during pregnancy is a major contributor to neural tube defects because of the critical role of folate in neural development. It is the key molecule in our cell, folic acid. And another problem is related to pyrimidine metabolism, hereditary pyrimidine 5 nucleotide deficiency. As I told you, this enzyme pyrimidine 5 nucleotase is able to use all monophosphate form of nucleotide, even if some chemicals it's possible for this enzyme. But in hereditary pyrimidine 5 nucleotide deficiency, this enzyme is used or specific substrate only use pyrimidine nucleotides. Hereditary pyrimidine 5 nucleotides 5' NT deficiency is most frequently abnormality of the cell nucleotide metabolism, causing hereditary non-spherocytic hemolytic anemia. This is a very important problem for patients. This disorder is usually characterized by hemolytic anemia associated with the accumulation of high concentration of pyrimidine nucleotides within erythrocyte. But we cannot understand real mechanism. It is not unclear for us. And when you look at pyrimidine degradation, we are a little bit lucky compared with purine degradative way, because uracil or thymine, of course, cytosine after that deamination reaction turns to uracil. Uracil goes to dehydroxyuracil, and we can open this ring system. It's so important for us, and produce an carbamoyl beta-alanine. And beta-alanine. Other side, thymine goes to dihydroxythymine and turn to carbamoyl beta-amyloidobutyric acid. It's possible to open ring system, and this molecule can turn to beta-amyloidobutyric acid. It is both molecules are soluble molecules compared with uric acid. For this reason, we don't have serious problem related to pyrimidine metabolism. But if you block the orthostat phosphoriboside transferase enzyme or OMP decarboxylase or UMP synthase, leads to orotic aciduria because of you block this enzyme must be increased substrate orotate increased in the cells. Patients with orotic aciduria excrete large amount of uric acid, but it's not so dangerous when you compare hyperuricemia. And then last topics is antimetabolites in purine and pyrimidine metabolism, because many molecules have purine and pyrimidine analogs and can be used as drugs. And remdesivir, for example, into triphosphate form, and this triphosphate form of chemicals it makes a complex with RNA-dependent RNA polymerase, viral RNA, and then inhibits the viral RNA synthesis. Remdesivir was the first drug to approved by the U.S. Food and Drug Administration for use in the treatment of coronavirus. The drug, which was used to emergency use in May, is recommended for use seriously ill patients. Acyclovir, also is purine analogs, is converted to triphosphate form, acyclovir triphosphate, and inhibits viral DNA polymerase and incorporate into molecular DNA and terminates the growing viral DNA chain and inactivates the viral DNA polymerase. Acyclovir is a nucleotide analog antiviral used to treat herpes simplex and herpes zoster and hepatic keratitis. Lamivudine is a pyrimidine analog, is associated with hepatitis B viruses. It is effective against potential. Lamivudine is a selective and specific inhibitor for viral DNA replication. It inhibits HBV DNA polymerase and terminates DNA synthesis and stop DNA synthesis and chain growing. And lamivudine is incorporated into the developing the chain. No other nucleotides can not to add the chain, and this results to inhibition of replication in the virus. As pentostatin, the purine antagonist antimetabolite, and that acts and other deaminase inhibitor and used in therapy of hair cell leukemia. After that, this information, I would like to complete my lecture on purine and pyrimidine metabolism. I hope to meet another lecture, mitochondria, electron transport chain and ATP synthesis, or replication, transcription, and translation lecture. Thanks so much. Have a nice time. Bye bye.